Inflammation is an immune response from the body when harmful stimuli or pathogens are introduced, and it causes the release of pro-inflammatory cytokines such as tumor necrosis factor alpha and the upregulation of enzymes such as cyclooxygenase 2. An alternative to the potentially harmful medications used to treat inflammation is treatment with extracts from plants, such as curcumin, which is extracted from turmeric roots and is known to have anti-inflammatory properties. Within this study, THP1 cells were differentiated into macrophages, stimulated to produce the immune response, and treated with varying concentrations of curcumin extracted by supercritical fluid extraction. A co-treatment, LPS and curcumin at the same time, and a pre- and co-treatment, pre-treatment with curcumin followed by a co-treatment with curcumin and LPS model was studied. It was hypothesized that curcumin extract would decrease the expression of TNF-α and the expression of COX-2 after co-treatment and pre- and co-treatment with LPS. To start, THP1 cells were grown in rPMI 1640 media, differentiated using PMA, and treated with both treatment groups. After treatment, they were analyzed using an MTT assay, an ELISA, and RT-qPCR. To make the treatment, curcumin was extracted using supercritical fluid extraction with an extraction pressure of 300 bar, CO2 at a flow rate of 1.5 ml per min, and a temperature of 35 degrees Celsius. Reagent alcohol was also used as a modifier. After SFB, the extract was rotary evaporated and resuspended in methanol. After that, the extract was analyzed using UPLC, using a C18 column and 0.1% phosphoric acid as mobile phase A and acetonitrile as mobile phase B. After treatment, representative images were taken to show fluorescence along with the cells under bright field. This allowed for the visualization of the treatment and to show that there was the same number of cells in each group, just more or less treatment. The images include the vehicle control and curcumin treatments of 1, 4, 8, and 12 microgram per mil. 2 MTT assays were completed to determine the percent cell viability after varying treatment concentrations within both the pre- and co-treatment and the co-treatment paradigm. Significance was shown in the pre- and co-treatment plate, suggesting that higher concentrations of curcumin significantly decreased cell viability. Using these results, the curcumin treatment concentrations were narrowed down to the 3 lowest concentrations for further testing. Treatments were normalized to the vehicle control because methanol is already known to be cytotoxic and all treatments contain methanol, so the vehicle control acted as a baseline. The delta-delta CT method was used for the analysis of CQ values obtained from RT-qPCR. A one-way ANOVA was created for both treatments with both expression of TNF-α and COX-2. While Tukey's multiple comparisons showed no significance within any ANOVA, the general trend showed the higher concentrations of curcumin could decrease the level of TNF-α expression. The general trend also showed that higher concentrations of curcumin could increase the expression of COX-2. This could be due to the cells being injured as the treatment concentration is higher, causing them to release more COX-2. The results from RT-qPCR were also normalized to the housekeeping gene, GAPDH, and compared to the vehicle control rather than the negative control. An ELISA was used to quantify the amount of TNF-α expressed after treatment. Results showed the general trend of a decrease in the amount of TNF-α after treatment within the pre- and co-treatment group. No significance was shown using Tukey's multiple comparisons. All treatments were normalized to the vehicle control due to methanol being known as cytotoxic, and all curcumin treatments were resuspended in methanol. Curcumin was successfully extracted using SFB and was analyzed using UPLC before treatment of cells. After treatment, MTT assays showed that pre- and co-treatment with curcumin and LPS significantly decreased cell viability as treatment concentrations were increased. Treatment of THP1-derived macrophages with varying treatments of curcumin in the pre-treatment with curcumin followed by co-treatment with LPS along with co-treatment alone with LPS resulted in a decrease of TNF-α expression and increase of COX-2 expression, as treatment concentrations increased, although no significance was shown. The decrease in the expression of TNF-α and the increase in expression of COX-2 should be confirmed using a larger number of replicates when completing real-time qPCR and ELISA to ensure significant results. Higher concentrations should be tested to determine if higher concentrations produce significance, since the concentrations tested within the study were all very low. When completing the ELISA, more dilution should be used for samples to ensure that they are all within the standard curve and can be analyzed. When extracting curcumin from turmeric roots in future studies, the extract should be rotary evaporated and resuspended in a solvent that is not cytotoxic, as methanol was cytotoxic and greatly decreased cell viability after treatment. Lastly, I would like to express my gratitude and appreciation to Dr. Jessica Livingston, Dr. Michael Gibson, Crystal McFadgen, Marla McKenzie, and Alea Quilty for their continued support and expertise throughout this research project.